Hello and welcome to this screencast where I'm going to take you through some of the genetic database activities that are required as part of the International Baccalaureate's diploma program biology course, specifically relating to topic 3 from the 2016 syllabus. There are 3 activities in this sequence, which all require the use of a variety of different online databases. If you like what you see, obviously please feel free to follow me on my Twitter handle at WRPVincent. Just a note about these databases that obviously for professional biologists these databases are an essential tool for the research they do. They store huge volumes of publicly accessible data that allow researchers and the public with the necessary skills to actually develop a deeper understanding of the processes of molecular biology, but also evolutionary relationships amongst different species and all manner of other areas. So we're going to look at 3 online databases in this sequence over 3 activities, but before we get there I just want to highlight that I have actually adapted these methods or this sequence and activities from the following sources. So I have relied on the work by Andrew Allott and David Mindorf from their textbook for the IB Biology course companion, as well as some of the work from the biology teacher resources on In Thinking, produced by David Farah. I think that's how you pronounce his surname. Please, David, correct me if I'm wrong. So without further ado, I'd like to get on to the first activity. So, hello there. In this first activity, this first database activity for topic 3, we're going to be looking at the first skill. The assessment statement for 3.1, skill 1 is use of a database to determine differences in the base sequence of a gene in 2 species. And in this activity I'm going to show you how you can use some information online to actually complete this task. First up, I should say that I've adapted this protocol from the Allott and Mindorf Biology International Baccalaureate Diploma Program textbook, where they do have step-by-step instructions on how to do this. I have adapted it slightly because I felt when I was preparing this for my students that this was a little bit tricky, so I thought just recording this screencast might make things a little bit easier. So several things you're going to need for this activity. First of all, we'll need to use the GenBank database online, and more on that in a second. First of all though, we'll need to install a piece of software known as clustalX, and we can use that to analyze the information that we gain from the GenBank database. So first of all, you just need to navigate to the clustal.org website. We're going to be downloading clustalX, so I just click on this link here, and on the page, the landing page here, I'm just going to go to the available for download link, and just looking at this list, making sure that I'm using a Mac, so I'm going to download the Mac OS X, but I want the clustalX program, not the clustal-W. Okay, so that's the clustalX program, and I'm going to, if you just click on this link, it will start a download for you, which you can install. Now, I've already installed this program myself, so I'm going to go to my Launchpad and just type in clustalX, and I'll launch that, so that's ready to run. Okay, so once we've installed that, the other program that we'll need to use on the Mac, and again it's Notepad on a PC I believe, but I'm just going to type in text edit, and I'm going to open the text editor. It comes up with this dialog box. I'm just going to click on new document, and then I'll be able to paste my nucleotide sequences that I want to analyze into this document, but first I need to save it as a plain text file. Okay, so just switch this over by going to in the text edit menu to format and then make not rich text. I've just done this, but make plain text. There we go. Okay, so now we're ready to roll. The last thing we need is some sequences to determine the differences of. So while this assessment statement does say the sequence of a gene between two species, when I run this, I'm actually going to run it with more than 3. Having played around with this myself, I found that there's actually a nice link to topic 5 when we look at cladograms, and if you've done more than 3 species here, you can actually use that information to create a phylogenetic tree for you. So you can save these files for use later on in the year. So next step then is to go to the GenBank database, which is just, you can Google GenBank. The link is given here at the top of my screen, and you'll get this landing page. Okay. We need to search for a gene, not a nucleotide, so I'm going to select this box and I'm going to scroll down. I've got a whole list of things here we need not concern ourselves with right now, and I'm just going to select gene. Okay, so down to the search menu and select gene. Now, following the instructions from the Allett and Mindorf textbook, I'm going to look for the COX1 gene across a few different species, and I'm going to start by looking for COX1 in Pan, genus name for the chimpanzee and the pygmy chimpanzee. So I'm going to click search now, and I should get a link or a list of the genes here that is available in the database. So you can see you've got chimpanzee and the pygmy chimpanzee and a few others. I'm just going to stick with the first one, COX1 in the chimpanzee. So I'm clicking on that link. This will take us to information about this specific gene. Okay, so this is the information about our gene. What we need for our purposes is under the genomic regions, transcripts and products. There's a link that says go to nucleotide. I'm going to click on the fast A, and this will give me a readout of the nucleotide sequence for this gene, and here it is. Now we need to copy all of this, so I'm going to just highlight all the information there, including the header at the top, and I'm just going to press copy. Okay. Now take that and we're going to paste that into our text file. Alright, and just to tidy things up a little bit, I'm going to delete the number here, and I'm going to put an underscore between Pan and troglodytes, and then delete this information. It will just make the display of our information in the clustr program a little bit neater. Okay, so I've prepared the one already, a bit like Blue Peter. So I've got my Pan, and I've got a few other species here. So the Neanderthal, polar bear, the modern human, the fruit fly, and the malaria-carrying mosquito. So once I've saved those sequences, as you can see, I've saved this file already. It's call it COX1. Okay, all you need to do to save in text edit, close, and then give yourself a name for your file and click save. I'm just going to delete this file because I won't be using it, however. Okay, so in clustr now it's open. I'm going to go to the menu at the top, and under File, I'm going to click Load Sequences. And then I can navigate to my COX1 file that's on my desktop, and I'm going to just click Open. And then we have some sequences here, but the last step is we actually need to align these sequences so the program can determine where the where the actual real differences are between the genes. So if I go to Alignment and then click, so that's in the clustr menu, Alignment, and then click Do Complete Alignment. This will give me an output guide tree. Now this goes back to what I was saying about topic 5. So this file could be saved for use later in the syllabus when we actually come back to look at phylogenetic trees and evolutionary relationships. So I'm going to press OK here to run the alignment. You can see it was working away there. And now this program cluster X has aligned our sequences. And this is where we have obviously the names of our particular species here. So we've got the Neanderthal and modern human. And now we can actually begin to see where the differences in this gene might lie between different species. Where there's a star at the top indicates that the nucleotide is the same. And so you can actually begin to look at how well conserved this gene is between different species. And scientists will actually use the number of differences here to map the evolutionary relationships or suggest how closely related certain species are. So that is it. That's all you need really to look at this skill for topic International Baccalaureate Biology Diploma Biology, topic 3.1, the skill 1 of using a database to determine differences in the base sequence of a gene of 2 species. Thank you for listening. If you like what you see, you can follow me on Twitter at my Twitter handle at WRPVincent. Thanks very much. Hello and welcome to the second database activity relating to the International Baccalaureate Diploma Programme's biology course for topic 3, genetics. In this short screencast, I'm going to demonstrate how you can use the Ensemble genomes online database to address the following 3 assessment statements from the syllabus, notably Application 2 in topic 3.1, which is comparing the number of genes in humans with other species, and then the second application in topic 3.2, which is comparison of the genome size in the T2 phage, Escherichia coli, Drosophila melanogaster, Homo sapiens, and Paris japonica, and then third application in 3.2, which is comparison of diploid chromosome numbers in the following species. I would like to highlight again that I have actually used 2 resources to help me develop this screencast. I've actually adapted the protocols given in the Allen-Mindorf biology textbook that is a course companion for the International Baccalaureate Diploma course in biology, as well as the biology teacher resources from the InThinking website there. So one thing that I give the students then will be this table that needs to be completed. Again, I have adapted this directly from Allen-Mindorf and combined it. They actually supplied the numbers from that in that textbook for these particular areas. I thought I'd leave these in as they might be particularly challenging. Some of these species are not included in the Ensemble database, although through a bit of Google searching you can actually find, for example, the number of genes for the T2 phage, obviously not the diploid number. However, on the side of being keeping things simple, I just wanted to look at one specific database which can be used to find the following information. So the number of genes for the following species, wherever there is a white box, that information can be filled in. So first thing we need to do is run to our Ensemble genome webpage. So you can Google Ensemble genomes. You could just type this into your browser. Now you'll notice that on the top right here we have these different tabs. So we have the database for the bacterial genomes, the protist genomes, fungi genomes, plant genomes, metazoan genomes, and then the vertebrates. And I'm just going to show you how to just search for one of these particular species, Homo sapiens. So wise man, modern man is a vertebrate. So I'm going to go to the vertebrate genome database, and here we have our Ensemble genome database. Now we can browse a genome. Popular genomes is here is human, but you can also select a species down here. So I'm going to go to the human via this route just to demonstrate that you can use this. You actually have a list of different species that you can see in this database. Okay, so we've got fish and amphibians, birds, reptiles, and mammals, and we're obviously going to go to the human and click through. So now we're on the information about the human, and here for all the species we see we have view carrier type. So I can click on this, it goes to go to human carrier type, click here, and there we have it. We have a carrier type image. Okay, so this will allow you, by looking at this, you can actually work out the diploid number of a species. And then if we click down through the database here, we can see that we actually have the information that we need for this exercise. So our base pairs, this is the total number of base pairs within this genome. So this is the genome size, and in the table from Alan Mindorf, this is given in million per million base pairs. So a short calculation can let you work this one out. So for genome size, we need to look at the base pairs, and then for the number of genes, we're going to look at the coding genes here. So how many actual coding genes are there in this specific database? And that's simple. That's really it. We can use that database then by going back to the over the top here. To look at the bacteria or fungi, whatever it might be, the particular species that we want to look at. So Escherichia coli will be in the bacterial area, Drosophila melanogaster is in the metazoan area, and so on and so forth. Thanks for watching. I hope that has been fairly straightforward. If you like what you see, you can follow me on Twitter at @WRPVincent. Hello and welcome to the third activity relating to the use of databases in topic 3, or genetics, of the International Baccalaureate's diploma program in biology course. I have, as with previous activities, adapted this activity from the Allett and Mindorf textbook or course companion, which provides some fantastic instructions on the use of these databases. This particular activity will address the assessment statement from topic 3.2, chromosomes, and it's the first skill 1, which is the use of databases to identify the locus of a human gene and its polypeptide product. In the activity for students, I also give them this table, which again is adapted from the Allett and Mindorf textbook, where I've removed the description of the gene, but given them 5 genes here which they can search for, where they can then write down what the gene does, what its polypeptide product is, and whereabouts on the chromosome in the human genome is it located. So this is a fairly straightforward activity. You can Google the OMIM, or the Online Mendelian Inheritance in Man database. I've hyperlinked it here, so I'm just going to go straight through to it. And the landing page, very clean, straightforward. You have this search bar, and then we have this gene map under the advanced search. Okay, so I'm going to first of all click on gene map on the advanced search section, and then you have this gene map advanced search, search the gene map, and also we have these chromosome numbers down the bottom here for the human genome. So firstly, just searching one of the particular genes, I'm just going to start with DRD4, which is the first gene in my table. I'm going to click search, and there we have it. We get this table printout that gives us the location of our actual of this gene, and it's on chromosome 11, and it tells you what this gene does. It creates it's gene for a dopamine receptor. So if I also click through here, it should bring up some more information on this actual genome, which may or may not be of particular interest. And that's it. It's very, very straightforward. I'm going to go back to show you one final thing. You can search obviously for other different genes here, and you can get the description of the gene and the location, the chromosome that it's found on. Or another search function we can do is just have a look at, we can select a particular gene, particular chromosome, sorry, and you can actually have a look at that. So I'm going to select the X chromosome, I'm going to click search, and then you actually have a list then of the specific genes located on that particular chromosome. Thanks very much for watching. I hope you enjoyed this or found it useful. If you like what you see, please feel free to follow me on Twitter. My Twitter handle is @WRPVincent. Thank you very much.